How many of you have heard the term gamete before? How many of you took health last year?
Oh, I did.
You've heard the word gamete before. Let me simplify it for some of you. I won't even make you learn it in Latin first. Gamete. Your textbook probably said some fancy thing about a specialized sex cell. In any mammal, you would recognize a gamete as eggs if you are a female and sperm if you are male. So a gamete is simply a sex cell. The cells involved with reproduction. Those cells have to come from somewhere. Autosomal cells are essentially everything else. Your liver, your kidneys, your skin, bones, muscle, those are all autosomes. Sometimes you'll hear people refer to them as body cells, which seems silly because gametes are also in the body. But they're not the reproductive cells. So anything that is not a reproductive cell is called autosomal. Anything that is a reproductive cell falls under the term gamete. Last chapter, you did mitosis, the process by which autosomes, autosomal cells, are made. They just make a direct copy of themselves, so a liver cell can make another liver cell. So a kidney cell can make another kidney cell. We do understand that mutations in that process are very bad, right? Like, it's pretty important that your skin cell makes an identical skin cell to what already existed. If that doesn't happen, what do we call it? It's a mutation which leads to particular kinds of diseases, such as cancer. That's where cancer comes from, is when a cell, an autosome, was supposed to make an identical copy of itself, and somewhere in that process a mutation occurred that causes it to divide over and over and over and over, which leads to tumors. That's what a tumor is. It's just a clump of cells that grow at a rapid rate. Are you prepared to teach this lesson? This is where I'll also remind some of you who don't have notebooks open or some note-taking device in front of you that you may want one. Gametes. How many of you have siblings? How many of your siblings are identical to you?
They're close.
No identical twins?
No.
How many of you are currently thinking of your sibling and now thinking, thank God there's not another one?
I wish I had.
There is a distinct difference. There is a distinct difference between producing an autosome and producing a gamete. The goal of autosomal replication, of those autosomal cells, is to make something identical. Your example of your siblings, it indicates, well, we know that people aren't identical. When two gametes fuse, the likelihood of making someone identical to someone who's already been made, that is actually impossible. Only one instance were there twins, identical twins, born 6 years apart. It happened during a case of in vitro fertilization. A mother was fertilized with an embryo and then 6 years later was fertilized with another one of her embryos that had been frozen for those 6 years, and it was an identical twin. It's the only instance in which that can happen.
Wait, how does it work?
How does what work?
How do you get froze?
because it's what you guys would probably refer to as a test tube baby. So there was a collection of eggs, ovum, gametes from the mother, a collection of sperm from the father. They were fertilized in a test tube, and usually in that instance about 15 embryos, 15 babies are made in that test tube. Actually, technically, even though they call it a test tube baby, it's in a petri dish, not a test tube. About 15 babies are made in that petri dish, and then a mother, at any time that she's fertilized with them, they usually only try and fertilize with about 5. Because usually with 5, one of them will implant and you have a successful pregnancy. There are a lot of instances in that case that you have multiple birds, and then they freeze the other ones. There are serious moral objections to this process for those of you who this is new biology to you. Very, very, very serious moral reservations to this. If you haven't already realized that that means that that human, that soul, got frozen for 6 years in a freezer, imagine spending the first 6 years of your life in a freezer. It's a person. Moment of conception, it's a person. There's a soul, right? I don't think that's up for debate. I don't think where you are that's up for debate. I'm telling you, it's not a debatable thing. I'm telling you the moment of conception, they receive a soul. Agreed? I can send you back to Miss Boileau if you want. Okay. So that's how. You fully recognize then that the goal of the division process is not to be identical. Right? Identical is usually bad when it comes to making new people. Identical? What if we were all perfectly identical? I'll make it a not people example for you so you understand why this would be bad.
Who would I be looking at?
A not people example, the banana you eat that you are familiar with from the grocery store is called the Cavendish banana. The Cavendish banana, every single Cavendish banana is genetically identical. They are all the same. Now here's the problem. There is the threat of the banana population being infected with a particular virus.
This is like the potato virus in Ireland. It just wipes it out.
Which means that if you haven't noticed the cost of the Cavendish banana going up, the reason that the Cavendish banana costs so much more money now than it did before is because the threat of all of those bananas being wiped out by one single virus exists because there is no genetic diversity. They're identical. So if anything can kill one of them, it kills all of them. So biologically, diversity is a really very good thing. Very good thing. So this process, this whole making of a gamete thing, is really biologically very important. But it's not that much different than what you did last chapter. I know because most of you started with a picture that looks something like How many of you chose that photo as your first photo? What does it look like to you? So if you're telling me it looks like interphase, if you're telling me it looks like interphase, what here indicated to you and your partner that that was interphase? That's a this is and that cell membrane. So we have one cell here, right? Just looks like a cell. What photo did you choose to be your next photo? So you looked for condensed chromosomes. How many of you are like, yes, that was the next logical place to go, condensed chromosomes?
Yeah.
Okay? Are you volunteering yours?
Sure.
How many of you say, yes, this is what we chose for second? What is it about this image that you would say this image is clearly the next thing after interphase?
The nucleus, you can see it hasn't or the envelope it hasn't disintegrated yet.
So we still have a nuclear envelope. But we also have, chromosomes, and we have, what did you say those were, Julie?
The centromeres, or the centrioles.
What would you label this if you were giving it a name? The one that comes after interphase? Prophase. Except, how many times does it look like prophase occurs in this particular set of diagrams you're looking at? Looks like it occurs twice. So we call this one prophase 1. Happens right after interphase, and it only happens in one cell. We started with one cell, we still have one cell. Now during that initial interphase, DNA did replicate. And there are some subtle differences here compared to what you saw last chapter. What looks different in this picture?
Looks like there are two of the X's.
There's two X's that are identical? Like two white X's and two black X's?
Yeah.
Okay.
There are more chromosomes. Like, does that mean that it splits more ways?
Not yet. But if you're saying that it looks different because when we looked at meiosis, did your chromosomes line up next to each other like that? Like, did they pair with their identical?
No.
So in prophase 1, when we're trying to make a gamete, the difference is we have what we call homologs or homologous pairs. They're homologs or homologous pairs. And they line up next to each other, and they pair. Now, the cell on your tabletop, I will tell you, has how many chromosomes total? 4. For the purposes of this example and my only having 2 hands, we're going to reduce that to 2. So this is what would have been inside the nucleus during interphase. Before S of interphase would have been this. After S, what happens to the DNA? Or during S, I should say. Replication. So there's how many of these then? So you end up with something that looks more like this, right? Now, again, I apologize like I did last chapter for my relying on gender stereotypes. For the purposes of making sure I turn that right on its head, this set's from Dad and this set's from Mom. They contain identical genes. So this set from Dad contains the hair color gene, where this set contains the hair color gene that you got from Mom. In prophase 1, the ones that contain the same sets of genes are going to line up next to each other, just like you see in that picture. They line up next to each other, and it's called a homologous pair. They contain all of the same DNA. And you'll notice I'm doing something with their arms. They cross. That's called crossing over. Something very strange happens, and it's difficult to think of it as far as like arms and legs goes, because we're going to get to anaphase, and something's going to happen when these get tugged upon and they separate. So it's like this. If prophase 1 came first, what comes second? If it goes in mitosis, it goes prophase, then? I appreciated, by the way, PMAT, those of you who actually wrote PMAT on the top of your tests in an attempt to keep them all in order. I like seeing how you're thinking. So what picture to you looks like metaphase? About that time, probably no sooner. Someone stole your metaphase.
This cornfield. What about this one that go like in between them?
So how many of you found a picture in between prophase and metaphase where it looks like the things are moving? There are some textbooks in this particular series of pictures included who would label the transition from prophase to metaphase as pro-metaphase. I don't expect you guys to know pro-metaphase so much as you know that there's like a series of things that must happen in between. Does that make sense? Is this just metaphase, or should we call it metaphase? So here's metaphase 1. Liam, you told me that you knew this was metaphase because of what? Metaphase plate, right down the middle. They're aligned at the center at the metaphase plate. Is this identical to metaphase, or is this slightly different?
Slightly different.
How?
It doesn't look the same.
What doesn't look the same? Are the spindles what's different here? Who thinks they can draw a representation of metaphase for me, of metaphase from mitosis?
They all just go in a line instead of. So.
She's an artist.
Oh, I was gonna use this side. Can I just circle it and show where it should go? So like, this one should go next to it, and this one should go next to it there, and it should be a line.
So in metaphase of mitosis, all of the chromosomes are aligned across at the equator. But here, they arrange at the equator as a homologue with their homologous pair, with their partner. They've aligned across the center. That's a pretty big distinction between metaphase and metaphase 1. So between mitosis and this process, the formation of a gamete. I haven't used that word yet, but yes. I'm trying to convince you guys that you pretty much already understand most of it already if you got last chapter. So it's not like all a whole series of scary things to learn over again. Are you convinced of that yet? Yeah, maybe. Well, let's see if you can impress me. What should come next then? Anaphase what? What should it look like? They're pulling apart. So they are migrating to opposite poles of the cell with one subtle difference. Did the centromere dissolve? So they're pulling apart whole chromosomes with the centromere still intact. So can you show me which one you think is your anaphase 1? I agree wholeheartedly. So we're looking at anaphase 1, where homologous pairs separate. Now, I will point out something strange here. Remember that whole crossing over thing we said that happened? Where the arm at top twists with the arm of another? As they pull apart, something can happen that we usually refer to as crossing over. Sometimes we call it disjunction. Keep in mind that, say, eye color gene. If this is mom's eye color gene at the very tip of this chromatid, and this is eye color gene at the tip of this chromatid. If they're overlapped, and in the process of pulling apart, they've twisted and you pull, it's possible for dad's chromosome to switch parts of arms. So it would be like your arms are interlinked with your partner or your, like, what do you put that ridiculous game you play, like Red Rover when you're a little kid, right?
yes.
And you have to feel like you're pulling hard. That's like if you pulled hard between each other, you would switch lower arms. Like you would have your partner's lower arm of the person standing next to you. We all agree that would be uncomfortable and weird. Or some of you are like, yeah, that'd be pretty cool. Okay? If that were true, that's what happens here. That crossing over causes a little bit of extra genetic reshuffling. Remember we said it was really important to be different? That ensures that we're a little bit more different in the gametes produced.
Does crossing over happen now?
Doesn't happen every time. They do line up next to each other every time, but they don't always exchange bits of DNA. Sometimes they exchange from like the elbow to the fingertips. Sometimes it's just fingertips, so the amount that they exchange. Sometimes it's not always on the arm, but it's on the leg, so it's like knee down or just the foot. So it depends. Every time it happens, it's unique, which ensures a little bit of extra guarantee of genetic diversity. Doesn't happen in anaphase, but it happens in anaphase 1. If you're going to follow anaphase 1 with something, what would you follow it with? Telophase 1. Follow it with telophase 1, what would that look like?
It's starting to furrow.
So we're going to see the formation of a furrow, and kind of looks like Pac-Man. How many of you are like, yes, telophase and cytokinesis in this particular set of daughter cells looks like Pac-Man? Now remembering there's a couple of extra cell pictures in there, so if you have one that occurs between it that looks like it has two separate regions of like gray DNA, that would occur between anaphase and telophase. How many daughter cells are formed after telophase 1?
Students, please excuse this interruption. Wanted to make a couple of announcements.